Hello students, this is CB Manoj from MIT College. Today's lecture is on chromosomal theory of inheritance. Hope so you have attended the last two lectures based on the same topic. Today is a continuation of the same. This chapter is basically based with the topics on heredity and variation. Today we'll be discussing on the structure of chromosomes. Now, before I start with the structure of chromosome, I would like to explain you the location or the position of chromosome in a cell. This is nucleus. In this nucleus, where is the chromosome exactly located? The position of the chromosome. This is a cell which shows presence of a nucleus. Within nucleus, chromosome is located. What is this chromosome actually made up of? What is there on the chromosome? It is nothing but the DNA molecule, which is a double-stranded coil structure made up of nucleotides. This is all we all know, the basic structure of DNA, which we people have learned in 11th standard. So where is the chromosome located? Finally, the chromosome is located in the nucleus of the cell. How does the chromosome appears to be? Now, if you split this word chromosome, chromo means color, soma means body. So it is nothing but a colored body. How can you call this is as a colored body? This is called as a colored body because of presence of a stain, a staining agent, which will give color to this chromosome. So it is called as a colored body. This chromosome is a thread-like structure which is self-replicating, and it is a genetic structure which shows presence of DNA molecule packed or found in the nucleus of the cell. Chromosomes which are seen during metaphase stage of mitosis are stained with the cells with the basic dyes, and they are visualized under light microscope. There might be a question arise in your mind that why the chromosomes are visible, are being taken only during this metaphase stage of mitosis. There are so many stages of mitosis, but why only metaphase stage? There is a reason behind this. In metaphase stage, the chromosomes are present in the equatorial position. So this chromosome which are present in the equatorial position, they are condensed chromosome. Because of the condensation of the chromosome or the short chromosome, they are easily stained by the dyes. And that's the reason why this colored body is seen under light microscope at the stage of metaphase of mitosis. And who is a scientist who came forward, who discovered a thread-like structure that appeared during cell division? There's none other than the scientist E. Strasburger in 1875. Now, in almost all types of higher organisms, that is eukaryotes, the nucleus, they contain definite number of chromosomes, and this chromosome has got a definite size and shape. Valdier coined the term chromosome for the first time, but Strasburger was the one who came forward with the thread-like structure. But chromosome, the name was coined by the other scientist Valdier in the year 1888. Now when you see chromosome, there are two types. One is somatic chromosome, other one is gametic chromosome. Somatic chromosome, somatic cells are nothing but the body cells. The cells in which the somatic chromosomes are present, which are diploid in nature, and they are represented by 2n, where this n stands for number of chromosomes. When talking about gametic chromosome, their chromosome number appears to be haploid. That is, they're just half of the somatic chromosome. So that's the relationship between somatic chromosome and gametic chromosome. Somatic chromosome is diploid, while gametic chromosome is haploid. When we talk about homologous chromosome, these are the morphologically, genetically identical chromosome. Now, if you consider in an offspring, the chromosomes are homologous, because amongst the homologous chromosome, the paired homologous chromosome, one chromosome is coming from the mother and the other chromosome is coming from the father. And those identical chromosome is nothing but homologous chromosome with respect to morphology and their genetic what is genetic constitution, that is gene content and the gene order. Now, there are two types of chromosome. One is autosome and other one is sex chromosomes. Autosomes are the one which will control the character of a body cell other than the sex characters, or which carries the genes for somatic characters. That is for the body cells. But sex chromosomes are the one which is involved in the sex determination. Whether the person is male or female is because of the presence of the sex chromosomes called as gonosomes. If you see in humans or most of other mammals, there are two types of sex chromosomes. One is X, other one is Y chromosome. So since there are X and Y, two different chromosomes present in humans or mammals, they are called as heterosome. If you talk about females, in case of female, the human females, there are two X chromosomes, so they are identical. But if you talk about males, there is one X and one Y chromosome. When I talk about birds, the female has ZW, so these are two different chromosomes, so they are heterogametic. And in the bird's male, it is ZZ, which is homogametic. This is just a correlation between X and Y. What is the difference between X and Y? Now, chromosome number. Chromosome number, as already I have explained it to you, it can be diploid, it can be haploid. Diploid is seen in the somatic cell. Haploid chromosome is seen in gametic cell. Gametes means the male gamete and the female gamete. For example, sperms and ova respectively. The chromosome number, they may vary from 4 to 1200. N is what? Gametic or haploid chromosome number. 2N is what? Somatic or diploid chromosome number. But why there is diploid or haploid chromosomes present? The diploid chromosomes are produced and haploid chromosomes are also produced by cell division. As you all know, there are two types. One is mitosis, other is meiosis. The mitosis cell division gives rise to diploid chromosome. So when you talk about meiosis results into haploid chromosome. Okay, the number of chromosome, it varies from species to species, but all individual of a species have the same chromosome number. All individual of a species will have the same chromosome number. Now, the number of chromosomes from species to species, it varies, but all the individuals of a species have same number of chromosomes. Closely related species usually have similar chromosome numbers. Presence of a whole sets of chromosome is nothing but called as euploidy. Whole set of chromosome is equal to euploidy. This euploidy can be N, that is haploid, it can be 2N diploid, it can be 3N triploid, it can be tetraploid, all S, etc. The one which contains single set of chromosome is called as haploid, which is represented by N. And the one in which in two sets of chromosome present, that is in somatic cell, it is represented by 2N, which is diploid. Now, triploid, tetraploid as it goes on. But there is one more term which is called as polyploidy. Poly means many, but what many? The multiple sets of N is nothing but called as polyploidy. Now, when there is any change in the chromosome number with respect to its normal chromosome number, that means either it might be addition or deletion of a chromosome number, it results into aneuploidy. That means it results into chromosomal disorder. When a change in the chromosome number does not involve entire sets of chromosomes, but only a few of the chromosomes, is called as aneuploidy. That means either addition or deletion of the chromosome set might take place, resulting into aneuploidy. It can be monosomics, it can be trisomic, it can be nullisomic, it can be tetrasomic. Means what is happening over here? As already explained, 2N means diploid chromosome. That is to a diploid set of chromosome. Here it is -1. So 2N -1 means diploid set of chromosome is reduced to 1 chromosome less. Here 1 chromosome is more added. Here -2. So 2 chromosomes are deleted over here. Here tetrasomic, 2 more chromosomes are added. So tetrasomic. That means there is a change in the number of usual set of chromosomes, that is aneuploidy. Now, next is the size of the chromosome. It varies. If you can see, it varies. It's as short as 0.25 micrometer in fungi and birds, but it is as long as 30 micrometer in some plants like Trillium. This chromosome, they have two arms. One is P and other one is Q. P is a shorter arm, Q is a longer arm. Okay, now next is the chromosome shape. If you can see, is usually observed at anaphase, when the position of the primary constriction centromere determines the chromosome shape. That means in case of anaphase, the centromeres, they are splitted. Each of those centromeres, sister chromatids, they reach the opposite pole where a spindle apparatus is attached to the the centriole at the poles to that of the kinetochore at the centromere, which we have already studied in cell division. Now, this constriction of the centromere is also called as primary constriction. They can be either terminal, they can be subterminal, or they can be the middle of the two chromatids, that is the median in position. Now, what is next? Something about homologous chromosome. What do you mean by homologous chromosome? Homologous chromosomes are nothing but which the copies of the chromosomes, they're identical in the morphology, in their gene content, in the gene order, that is genetical content, is nothing but called as homologous chromosome. It is a diploid organisms in which they have two copies of each chromosome. That is, one chromosome is split into two, except the sex chromosomes. Diploid organisms, they have got two copies of each chromosome. And each pair of the chromosome is called as one pair. Each pair of the chromosome is called as homologues. So in a homologous chromosome, there will be two homologues in a paired homologous chromosome. Now, if they are saying that they are paired, so one chromosome in homologous chromosome is from mother and other homologue is coming from the father. If you can see over here, there are two chromosomes. This is one pair, this is a second pair. So they have, how many pairs are there? You can call this as not two pair. You can call this homologous pair. You can call it as homologous chromosomes. You can call here it as. Now, this one chromosome might come from parent or from mother or from father or vice versa. And this might come from father or mother. Now, in this case, as you can see, the tip here end is telomere. This one is a short arm in both the cases, and other one is a long arm. So these are the sister chromatids. This is one chromatid which is split into one more. That is, before cell division, all this splitting takes place. And this one chromatid is linked with the other chromatid by means of a center point, which is nothing but called as centromere. So these two chromatids are nothing but called as sister chromatids. Even over here, the same. These two chromatids are called as the sister chromatids. Okay, and this is one X chromosome and this is the other Y chromosome. Okay, now, structure of chromosome. The structure of chromosome is usually studied during the metaphase phase of the mitosis. During metaphase, the chromosome is usually more condensed form so that they can be easily distributed to the daughter cells. And when you talk about the chromosome in the interphase stage of mitosis, here you can see the chromosome, they are thin, they are coiled, elastic, and thread-like structures. That means the chromosome structure, it varies from one stage to the other stage of the cell division. In mitotic metaphase chromosome, the following structural features can. Now, what type of structure is chromosome? If you see the structure of chromosome, they have the following parts, such as satellite. This is the arm. This is a centromere or the primary constriction. This is a secondary constriction. This is a telomere. These are the sister chromatids. Okay. Now, in this case, each part of this structure will be discussed in the later slide. You can see over here, chromatid. Now, I have taken the same structure of chromosome. Let's explain each structure of this chromosome. Chromatid, each metaphase chromosome appears to be longitudinally divided into two identical parts, each of which is called as chromatid. If you can see this structure, this one structure, this is one chromosome. There's a duplication of this chromosome and it is over here. But when this duplication is done, when this replication is done, multiplication is done, these two chromosomes, they are linked by means of centromere. So this is called as one chromatid, this is the other chromatid. So these two chromatids, they are the sister chromatid. And if you have like this one more chromosome over here, so these two chromosomes will be homologous chromosome. And their chromatids are non-sister chromatid. But here these two of a particular chromosome, they are sister chromatids. And these chromatids, which are longitudinally distributed, they are held together by means of a centromere, which is called as a primary constriction. Okay, now the two chromatids of chromosome separate from each other during mitotic anaphase. Obviously, if you remember the cell division, in its anaphase stage, the two, now from here, the separation of this chromatids takes place. One part will go to one pole, the other part will go to the other pole, and where the centrioles are present. And from there, this chromatids are held to the, in the, what do you call, in the center by means of mitotic spindle. That means it is held to from one pole to other pole, it is held by mitotic spindle. Since the two chromatids making up a chromosome are produced through replication of a single chromatid during synthesis phase. If you remember, synthesis phase is a part of cell cycle before the mitosis takes place of interphase, they are called as sister chromatids. That means this is a sister chromatid. This is one chromatid, the other one. So both are sister chromatids because this chromatid is formed by presence of this replication of this chromatid. So both these chromatid are nothing but called as sister chromatids. In contrast, the chromatids of homologous chromosomes are known as non-sister chromatids. That's what I said. So this is one chromosome. I need to take one more homologous chromosome. So if you consider the chromatid of this and one more homologous chromosome is present over here, so the chromatid of this and the chromatid of this are called as non-sister chromatid. There's a difference between sister chromatid and non-sister chromatid. Now, what is this? Again, I'm showing you the same diagram to get rid of the confusion. Centromere, which is called as a primary constriction. That's what. This is a centromere, also called as a primary constriction. Each chromosome has a constriction point called as centromere, which has got one more name, kinetochore. This kinetochore, this will divide the chromosome into two sections or arms. Now, because of centromere, this chromosome has got two arms. One is short arm, other one is long arm. Short arm is represented by P arm, long arm is represented by Q arm. Okay, now the next part is telomere. What is this telomere? The two ends of the chromosome are known as telomeres. They are involved in chromosome replication and the maintenance of chromosomal length. These telomeres are highly stable. And because of these telomeres, the chromosomes do not fuse with each other, but they are in contact with a nuclear envelope. This telomeric region is made up of repetitive sequence of thymine and what do you call guanine bases. These are T and G. This you can see over here, T and G. This is thymine and guanine bases of nucleotide bases of DNA. The next one is on secondary constriction. What is secondary constriction? Secondary constriction in some chromosome, addition to centromere or primary constriction, one more constrictions in the chromosomes are seen, which are called as a secondary constriction. You can see here, this one chromatid is been again connected by means of secondary constriction. The way it is shown over here is also over here, also here, also here. Okay, this is a secondary constriction. Satellite. What is satellite now? The chromosomal region, see here, the small chromosomal segment separated from the main body of the chromosome by a secondary constriction is called a satellite. A small is a small chromosomal segment which is separated from the main body of the chromosome by a secondary constriction is called as satellite. The chromosomal region between the secondary constriction and the nearest telomere. The chromosomal region between the secondary constriction and the nearest telomere is called as satellite and chromosomes. That possess this region called as satellite chromosome or SAT chromosome. You can see here a satellite is present over here. So the chromosome in which this satellite is present is called as a satellite chromosome or SAT chromosome. Now what is next? Next one is the centromere position. The size of the chromosome. The size of the chromosome, it varies from stage to stage of cell division. The chromosomes, they are the longest and thinnest during interphase or the resting stage. Hence, they are not visible during light microscope. While the chromosomes, which are the smallest and thickest during mitotic, is mitotic by metaphase. That means here we can compare during two stages. One is an interphase, other one is metaphase. In interphase, the chromosomes are longest and thinnest. And in metaphase, the chromosomes are more condensed. Okay, so the size of the chromosome, it varies from one stage to the other stage of the cell division. And the chromosome size is not directly proportional to number of genes present in that particular chromosome. The location of the centromere on each chromosome will give rise to different shape of the chromosome. Where is the centromere located on the chromosome? That location will give a particular shape to that chromosome. Now, what is next? Chromosomes, they are classified according to the centromere position. As already mentioned, where is the position, where is the centromere present will be the different types of chromosomes based on the based on the position of the centromere. If the centromere is present at one end of the chromosome, it is acrocentric. If the centromere is present closer to one end, it is submetacentric. If the centromere is present in the middle, it is called metacentric. That's the difference between the three. Now, each of the chromosome, as already specified, they are made up of two arms. One is a P arm, other one is a Q arm. P is the shorter, the name is for petite. Petite means small. And Q comes because P is been followed by Q alphabet. Okay, so this is about the different types. This will be explained in next slide, next lecture, the types of the chromosome based on the centromere position. Now, as you can see over here, this is, I have shown just a small structure on, this is unduplicated chromosome, this is duplicated chromosome. Unduplicated means what? There is no duplication. Now here one chromosome is shown in blue, other is shown in purple color. Can you see any one more blue color over here? No. Can you see one more purple chromosome over here? No. But if you see here, this is a duplicated chromosome. In this blue color is again duplicated. This purple color is again duplicated. Fine. Now what has been seen over here? That is this unduplicated chromosome. This is duplicated chromosome taking place. Fine. Now these are chromatids. Already showed it to you. Okay, these are chromatids. But when a duplication takes place, this and this are the identical sister chromatid, and this one blue color and one purple color chromatid, they are non-sister chromatids of homologous pair. So these are the chromatids. These are the P arm, this is the Q arm, and the middle one which connects the chromatids are nothing but the centromere. Okay, now the next lecture will be based on the different types of acrocentric, submetacentric, all those types of central chromosome based on the position of the centromere. Thank you.